the seminar. Welcome to GSI seminar series. I'm Alla Grishok, co-director of GSI, and I'm responsible for organizing the seminars. Today, our guest is Dr. Yana Bromberg. She's an associate professor, Department of Biochemistry, Microbiology at Rutgers University. She also holds an adjunct position at the Department of Genetics at Rutgers, and she's a fellow of the Institute for Advanced Study and at the Technical University of Munich, Germany. Yana is also the vice president of the board of directors of the International Society for Computational Biology. Dr. Bromberg received her bachelor degrees in biology and computer sciences from State University of New York in Stony Brook, and a PhD in biomedical informatics from Columbia University. Research in Dr. Bromberg's lab at Rutgers is focused on designing machine learning, network analysis, and other computational techniques for the molecular function annotation of genes, genomes, and metagenomes in the context of specific environments and diseases. And this, of course, includes human genome and also microbiomes. And this research is funded by NIH and NSF. The lab also studies evolution of life and the selection of electron transfer reactions in Earth's history as a potentially applicable to other planets. And this interesting research is funded by NASA. Dr. Bromberg's work has been recognized by several awards, including the NSF Career Award, the PH Arma Foundation Young Investigator Research Starter Award, and the Hans Fischer Award for Outstanding Early Career Scientist. I must say that Dr. Bromberg is a leader in her field. For example, she has been chosen to chair a Gordon Conference on Human Single Nucleotide Polymorphism and Disease from 2020 to 2022. She is frequently invited to talk about her research in conferences all over the world and has to date co-authored more than 70 peer-reviewed scientific articles. Today, the title of Yana's talk is Decoding the DNA Blueprints of Human and Microbiome Molecular Functionality. Welcome, Dr. Bromberg, and we are looking forward to your talk.
Thank you so much, Alla, for the invitation to speak and for such a wonderful introduction. And thanks everybody for joining. I know that these Zoom meetings are getting on everybody's nerves, so thank you for being here. Let me share my screen and we can start. Are we sharing?
Yes, everything is working.
Okay, so I'm going to talk about, hopefully, if I get enough time, human and microbiome molecular functionality. So before I start, I ran into a couple of illustrations that I think are really descriptive of the state of the field right now. Sorry? Okay. So the idea here is that if you go and you listen to a talk, usually what you would like to get out of it is some correlation between the amount of time you spent listening and the amount of understanding on the talk that you actually have. So usually you'll start out at about 100%. Hopefully you're not lost by the first 5 minutes, and your understanding of the subject never drops below 50%. And hopefully by the end of it, you walk out with about 75% of what the person was talking about. Now, the typical situation is kind of like this. So you start out okay, then you drop to 0% understanding and you leave with 0% understanding. So I'm going to really hope that this is not the case here. And to accommodate that, I encourage you to speak up if you don't understand something. So just interrupt. I'm not going to be seeing hand raised or any chat messages. So please simply interrupt and let me know that you didn't understand. Okay, so my lab studies function, molecular function. And I know that means different things for different people, but from my perspective, these are the things that are involved. We study genes, genomes, and variation in those, protein structure, protein function, and the resulting systems. And today I'm only going to focus on the genes and genomes version of this stuff. We do this all in a dry lab, so everything is done computationally, hence the computers here, and this little artificial neural network that looks like a brain on this side. Okay, so what are we gonna talk about first? I normally talk about the genome stuff first, but I decided to switch it up a bit, and we're gonna start with the microbiome story today. So everybody knows that microbes are everywhere. You have them in soil, you have them in the oceans, in the rivers, and you have them in animals and in humans. And these microbes are not just passive inhabitants of the world that they live in. They do a lot of things that contribute to the activities that we attribute to all of these, so definitely to the animals and to the fertility of soil or to the richness of the ocean. So the problem here is that assessing the microbiome members is very difficult. In order to figure out who is there in the microbiome, you need to sequence the bacterial genomes in the sample, then cut them up into small pieces, and then try to reassemble them to make sense, to come back to this set of genomes that you're looking at here. Now, just to sort of bring it down to a more understandable level, this is my favorite library. It's in the Trinity College in Dublin, and it's beautiful. Now, if I asked you to tell me what books are there, just like I would ask who is there in the microbiome, what you would do first is shred every single book. Then you would take a little tiny scanner and you would scan the little pieces that you got, and then spend God knows how much time on really expensive compute equipment in order to be able to recompile the little shredded pieces that you've put together, that you've scanned, to put together the books that were there. And by the end of this, it's probably not a surprise that you will get only a small fraction of the books that were there. And you will probably get the books that were most represented. Now, I'm not saying that the Trinity College library is full of Treasure Island books, so Les Miserables, but this is probably what's going to happen with this particular experiment. So it's important here to remember that who is there, like what are the books, is not as important as what is actually happening, what are they doing. So who is there? This is a wonderful illustration that I found online, attributed to this Mouthville person. He draws or she draws a lot of cartoons on this topic, but basically these are the inhabitants of Mouthville, which is a microbiome of the mouth. You have Ginger, Fred, Justin, Katie, Jasper, and Barf the dog. Now, knowing who is there is not going to describe what Mouthville is doing. Actually, knowing what they're doing, like chemotaxis and invasion of epithelial cells and carbon fixation, that stuff will actually tell you what they're doing, right? So how do we know this? And so my lab designed this method called MyPhaser. I'm going to tell you really briefly how MyPhaser works. So imagine that you have a couple of genomes that have annotated genes with known functionality. The functionality here is represented in the sense of EC numbers, which is basically an ontology that tells you at different levels of gradation the precise function of a particular protein that this gene encodes. So for instance, anything that has a number 1 in the first position is going to be an oxidoreductase. The following numbers are going to be describing the substrates and the bonds that are being acted upon, so that by the end you have a very precise definition of what a particular protein is doing. So for instance, 1.2.1.1 is an oxidoreductase that acts on aldehyde or oxo groups of donors with NAD+ or NADP+ as an acceptor, and is a formaldehyde dehydrogenase. So these annotations are available from experimental studies of the completely sequenced bacterial genomes. And we wanted to use this information in order to develop a method that would help annotation of microbiomes. So let's go with these genomes and we say, okay, we can, if we were to sequence one of these genomes at random, we could have reads that have portions of the gene that's that number and portion of a gene that's another number. And then here's one more that doesn't have annotation and one more that has only the one number and so on and so forth. So once we've cut up this genome, we can do the same with another genome. So from this point, we can proceed here, which is basically the MyPhaser algorithm. So we take the reference proteins from these genomes that we just cut up, from these organisms that we just cut up, and we look at them versus the reads that we've just gotten out. So we translate the reads, we use 6-frame translations, and we align these reads to the proteins that are in the genome, are encoded in the genome of a different bacterium. So you see, this is a red 1.1.2.1, and this is a blue 1.1.2.1. And those two things carry the same functional annotation, so that's considered correct. That's this blue circle over here, while these two, for instance, carry incorrect annotations. So by doing this, we are able to place these alignments of reads to protein sequences into a two-dimensional space of alignment length versus sequence identity. And here we see that most of the correct values, so these, remember these blue values over here, tend to fall towards the top and to the right of the plot, so longer alignment length and higher sequence identity. And the red buttons, so the red circles over here, this is the incorrect ones, tend to fall on the left and to the bottom. So what we were able to do is optimize the separating curve. So this curve is, we called it phaser, which stands for functional annotations of sequencing rates. And the distance from this curve up along the sequence identity axis actually decides how reliable a particular prediction of this read having the particular annotated function is.
Yeah. But like at the same time, he had ice on his nose that was hurt. So like.
Okay, sorry. So the
Sean, there is interference from you, Sean Lines.
Okay. So the performance of this method, you don't have to go too much into the detail of this, but this is a precision recall curve. And you can see that the performance of this method is actually significantly better for known reads, the reads that we know, than, let's say, BLAST, which is a favorite of many people in order to try to figure out what is the particular functionality of a sequence. So we built a pipeline that you're welcome to use. Basic idea is you just throw in your metagenome sequences, and actually fairly fast after you should get an answer. And this answer looks like this. I'm sorry if you're not seeing it very clearly, but this is a list of functions and their abundances, right, in this particular world. So you've sequenced this metagenome, and these are the number of reads that map to a particular functionality. So why is this important? Well, we've represented the metagenome as a vector of molecular functions, and we can compare metagenomes in order to be able to establish certain specific features.
Yana, could you mute yourself? I muted everybody.
Here we go.
Sorry about that.
No worries. Okay. Did you guys hear me about these guys, the sick people? No. Okay. So we have a family that has Crohn's disease in the family. So these two people in this family have Crohn's disease. Everybody else is healthy. And what we were doing is we were comparing the functional vectors that we had identified using this tool, MyPhaser, that we had. And we see very clearly here, so this is a tree representing the distances of these people from each other on the basis of their functional similarities. And what you see is this SO1 and SO9. These are these two sick individuals over here are actually grouping away from the rest of the family. And they are also fairly far removed from each other. So the length of the branch here indicates that they are not very similar, right? So if you look at the healthy people, you further see separation. So for instance, the first one here are these husband and wife team over here in the older generation. So this is the first generation for which we have the data. And they have a very similar microbiome, as you can tell by the fact that their branch lengths are very small. So they're grouping together. The curious part is that they're also grouping together with this person. So this is a third generation kid over here that seems to have a very similar microbiome to these two people over here. And that's a kind of a strange situation, except if you consider that in the light of the fact that the mother of this child, S12, is actually here, right? And is grouping with SO3, who is the child of this pair, right? So there is some sort of transiency in the functional abilities recorded but for the different people. So these are people living in different households, yet S12 and SO3 are very similar to each other functionally. So SO3 and S12. These also happen to be women of the same generation, and we see the same signal for women or girls of another generation. So here, these guys are similar as well, S10 and SO5. And we also observe that SO6, this is the father in this family over here, is very different from everybody, but he's sort of in the mix over here. And the most different person in the healthy group is this SO4 person who happens to be an infant. So this is a child, and their microbiome is extremely different from everybody else, but it's even more different from the sick individuals over here. So this is something that indicates that we can actually look at the microbiome of the individual patients or people, sorry, and define whether they're going to be patients or not. But as you can tell, since the Crohn's disease patients don't actually look the same, they don't necessarily have to be alike to each other, right? Healthy people tend to be more alike. So if you actually look at what's happening in terms of the pathways that are upregulated in these two people that are sick, you see that there is lipopolysaccharide biosynthesis, which is known to be involved in promoting inflammation in bacteria. And it's also surviving, so the glutathione metabolism, which is known to help bacteria survive inflammation. So it's actually a very informative way of looking at what's happening in the microbiomes. We also looked at obese children that have Prader-Willi syndrome that were placed on a diet, a very particular diet that wasn't food limiting, but was the type of food limiting. And what you see very much is that between the children, so each box over here represents the functional diversity of the kids. So basically how different are two kids in one date. And what you're seeing is that at day 0, they're all very similar. So the diversity is limited, but as time proceeds over the exposure to the diet, they tend to grow more diverse. which is actually a good thing in this case. They also lost a lot of weight, which was the whole point of this thing. So that's another way of looking at it. And here's one more thing. So we do machine learning. And of course, if you get a vector of numbers, which is what the abundances or the numbers of functions are per particular microbiome, and a label, you can try to train a model to recognize whether particular signatures of function actually indicate a particular phenotype. So in this case, we had microbiomes from 2 type 2 diabetics, and we were able to train a support vector machine to recognize whether a person has type 2 diabetes or doesn't have type 2 diabetes. And this is not a very amazing story, because as you can imagine, type 2 diabetics usually change their diet to accommodate that. So you would see that microbiomes will shift from what you would expect in a healthy individual. But what we found very curiously in this thing is that the functions that are most enriched or most differentiating these 2 types of people, so type 2 diabetics versus healthies, are these 2 pathways. So pentase phosphate pathway and prokaryotic carbon fixation pathway, which are both involved in sugar metabolism, in bacterial sugar metabolism. So this is actually an interesting finding in itself as well. So all of this is cool, but we are somehow limited when we're using MyPhaser to the things that we can pick up by aligning, right? So by homology. And that's a big limitation, believe it or not. There are things out there that are not looking like anything we know. So if we wanted to study something new, we would have to use a different method. So very recently, I think this was in December, then we had put a preprint out. My postdoc had built a language model. This is the rage recently, deep learning. It's not that we really had to do deep learning. It's that we wanted to. We felt this was the right question for it. So this is not following trends. It's just simply the right question. And so the idea of a language model here is to try to create a representation of a read rather than in letters, to create it in some multidimensional space that captures the context of the entire sequence. So you know how a read is a sequence of letters? In this multidimensional space, you can create a different representation of every read. And so in order to create this model, what we did is we would mask out every nucleotide and try to predict it. So you would train the model by trying to predict the next nucleotide in sequence. So this is pretty stupid, not a very important thing to do. And it doesn't do very well. It does better than random, right? So it's about 40% accurate. So random would be whatever the distribution of nucleotides is. But the idea is that we wanted just a representation. So something as meaningful as a letter to the computer. So not a very meaningful thing, but something that would capture the context in which this letter appears. And what we found is that once we had that model, we could train models on top of that would do things that we really wanted to do. So for instance, we are able to make predictions of the EC numbers the way that MyPhaser does. We can do it without alignments by just taking the read and saying this is the function of this read on the basis of this encoding that we had created. So here, what you see is this is the first EC digit. So remember, there are 4 EC digits. I just showed 3. There are 4 EC digits. And the first EC digit is the biggest one. So the oxidoreductases versus hydrogenases, for instance. And what you're finding here is that this is actually fairly accurate. So it's no less than 80% accuracy in predicting that EC digit, and actually fairly accurate for second or third. And I'm not showing you the fourth, that just doesn't look good. But the total performance is about 82% accuracy. So you would say, okay, so that's a better mailbox. It's a faster way of identifying the same functions, right? This is the things that we could identify with alignments. Well, here's a cooler thing that you can do. Let's say I didn't want to know what the exact function was, but rather I wanted to know whether something is or isn't an oxidoreductase, right? So that would be my question. Well, I can train a model to do that. And what you see here is the performance of this model. So it can label 100% of the reads, obviously, as all machine learning can. And it identifies 20% of all the reads in this metagenome that we were looking at to be oxidoreductases. If I use my phaser, you can barely see any of the oxidoreductases it identifies. My phaser is very precise, but it's not going to give you a lot of annotations because you need that alignment. Here's mgrest, which is not very precise. It's a lot looser. People use it a lot, right? But in the end, it's also unable to perform as well as this. And one, you can ask me then, well, how do you know that these are correct predictions? I can give you a prediction of anything, right? I can annotate anything you want. How do I know it's correct? Well, we actually evaluated the biological evidence that we had for this being correct. And I'm sorry, this is really tiny now that I'm looking at it. But the idea is that this is the percent of oxidoreductases mapped versus the location of the metagenome that is being evaluated. So OMZ stands for oxygen minimum zone. This is this halfway on the from the surface to oxygen minimum zone and surface. And what you see here is, as expected, you expect a lot fewer oxidoreductases on the surface, and there are a lot fewer oxidoreductases on the surface than in the oxygen minimum zone. And this trend doesn't exist for either mgrest or myphaser. So not only is this particular classifier able to recognize things that other methods cannot recognize because of the limitations of homology, it's also able to do it correctly, to understand things that are not really well encoded by other methods. Okay, so some conclusions. Metagenomic functional differences across samples are important. So it can actually tell you whether someone is sick or why that person is sick, or what are the functions of the microbiome that are relevant to them being sick. And myphaser, our method, and soon this really cool deep learning myphaser, accurately highlights conditionally important functional pathways. So that's the microbiome story. There's a lot more to tell, but I wanted to switch into the genetics. Okay, this guy is an inspiration. He is a Hungarian-American mathematician. This is John von Neumann. He wrote one of the later papers in his life. I want to say one of the last, but it's not the last one, so I can't remember the order. But the paper was probabilistic logics and synthesis of reliable organisms from unreliable components. And now, if you get carried away with that, this is not about organisms per se. This is about automata, so computational paper, right? But the idea applies very well. So you could think of error not as an extraneous and misdirected or misdirecting accident, but as an essential part of the process under consideration. And when I talk about error, I talk about genome variation. So I'm going to start with talking about non-synonymous SNPs, right? So these are the single nucleotide substitutions, which result in an amino acid change on the protein level. And as you can see here, this is a thing we put together fairly recently. There's over 180 computational methods that try to predict the effect of non-synonymous SNPs. So one of them is mine. It's called SNAP. It's been around for a while. In my defense, it's been there when there were only 2 other ones on the market. So mine was in the first 3 or so. So the question that I like to ask my students when I talk about this, I say, why do we have so many methods to do the same exact thing? And the answer usually is because none of them does well. And that's the point here, that actually that's probably true. None of them is able to identify exactly the same things. And so if you look at their evaluation, I hope you guys are familiar with the ROC curve story, right? So you cut off the test score and you figure out your rate of false positives and true positives at that test score. And the goal here is to be as high as possible as a curve. So the higher you are, the better performing you are. Right? So the idea is that when you display how well your method does, you have to evaluate how well you can predict effect versus no effect of a variant for an available experimentally determined data set, right? And then you have to say that you do better than somebody else, right? Or everybody else, however you want to define this. So whenever you read these papers, you see these curves. So this is out of the Polyfen paper, and Polyfen people are your neighbors over at Harvard. And the idea here is that Polyfen, this green line, is higher than Polyfen 2, which is the green dashed line, but more importantly, higher than SIFT, which is in purple, and higher than SNAP, which is in blue, and higher than SNP3D, which is another tool. So that was the performance of Polyfen. Polyfen did really well, and just for a little bit more explanation, the shape of the curve and how far up it goes actually tells you what is the overall performance. So not just, how far above is it, but also what is the area under the curve for this one. So here's a cool thing. This is SNAP performance. So SNAP2 in this case, outperforms the original SNAP and SIFT and Polyfen 2. And of course, the moment that we publish that paper, there's going to be another paper. There's going to be a CAD paper that says that we outperform everybody else as well. But look at this. They're all about the same height. They're all about the same performance. These methods are about the same. So everybody could be outperforming everybody. But if they're doing it by a tiny little bit. So something is going on. And I think the important part here is to remember that protein variation effects are not 0 or 1. It's not, yes, there is an effect versus no, there is no effect. It's a range. So here's an experimental mutagenesis result. So basically, people took 2 proteins and they evaluated the effect of the mutations that these proteins had and the stability of binding to their substrates. So if you are at 1, that means that you are no effect. If you are this way to the right of 1, that's a gain of function. If you are to the left of 1, it's a loss of function. So in other words, that's more stable, that's less stable. That's the idea here. And what you see very clearly is a distribution. That's the first statement. You see a distribution. It's not a 0 or 1, right? And nonsense mutations, which you expect to always have an effect, actually don't. So they're a distribution. This red thing here that goes all the way down to 1.4, which is a gain of function, is actually the nonsense mutations. The yellow thing over here, the synonymous variants that everybody says has no functional effect on the protein because it doesn't change the protein sequence. Actually, they're distributed from 0.2, which is a lot of effect, to all the way up to 2 over here, which is a gain of function, right? And then the non-synonymous variants actually behave as, finally, something behave as you would expect it to. It actually distributes as a mix of these two. Right? So what we did is we used some mathematics to represent the non-synonymous variants as a mixture of 2 Gaussian based on the synonymous variation in the dashed line over here and the nonsense variations in the solid red line. And what we said is that according to this, as long as the probability of it being more nonsense-like is less than being more synonymous-like, then this thing has no effect. Otherwise, it has an effect. So the cutoff where these 2 lines cross, the dashed and the solid, is where we said that we're going to say there is an effect. But again, I want you to remember that the effect is actually a distribution. Okay, so we choose this cutoff, and then we ask these 2 predictors, Polyfen and SNAP, to make the prediction of effect versus no effect. So we compared the experimental score to the SNAP score, and we transformed the experimental score to mean at 0, you have no effect, and at 2, you have a lot of effect. So we just basically just caused the transformation. And what you see that there is that there is a correlation of no effect to of effect to a SNAP score. So there's this red line over here. So SNAP score from no effect to effect, experimental assay from no effect to effect. Same thing we did for polyphen. So from 0 to 1, that's a polyphen score, and the y-axis is the experimental score. And there is indeed a correlation. But here's a story here. The correlation for polyphen is actually driven by a bimodal distribution of variant effects. So basically, polyphen mostly predicts them to be 0 or 1, and not enough of them in the middle. On the other hand, SNAP scores are normally distributed. So the idea is that the polyphen predictor is actually going to say yes or no. And depending on your cutoff, you may get it right or not. So according to our cutoff, it did well, right? But we don't have the data of an experimental mutagenesis to try to figure out whether something is an effect or no effect. So I just want to show you that if I only focus on predicted SNAP effect scores, and only the ones that have an effect according to our cutoff, then the correlation becomes even better. So the correlation between the amount of effect that is reported by an experimental assay and the SNAP score is actually fairly strong. But this is not surprising to us. We knew this. When we looked at the data set which we used in training of SNAP, we saw that there is a difference in predicting severe, moderate, or mild effect variants according to the effect. So I put the curves in, they're not actual curves, but it's just to make looking at this data easier. You see that severe variants, experimentally annotated severe variants, tend to be more right-shifted than the moderate variants, than the mild variants. But what is curious about this distribution here is that neutral variants, or experimentally annotated variants that carry no effect, are actually not neutral. Well, halfway are not neutral. And I mean, those of you who are experimentalists probably understand why. Because in order to prove that something is experimentally neutral, that doesn't have an effect, you have to do 5 million experiments in every possible condition, right? But to prove that something has an effect, all you got to do is one, right? So all you got to do is show that there is an effect in one case. So the neutral variants in literature are usually just like a byproduct of doing some kind of experiment and not seeing an effect for this particular variant. But it doesn't mean that there is no effect at all. And so I bring this up for a reason. So we evaluated the variance in 1,000 genomes data, which includes a lot more than 1,000 genomes, but this is human genomes from healthy individuals. And what we saw is that actually the distribution of the variance scores, this dashed black line, is even less of lower than the experimental neutral line. And also more left shifted, so there is this thing over here, then the distribution of scores for HapMap data. Now, HapMap data, these are the common variants. So 1,000 genomes data includes all human variants. HapMap data includes only the common human variants. Now, it's general practice in medicine to assume that common variants do not contribute to anything, to any disease or any other issues, except as you can see here, they have on average more effect than rare variants. And this is not something that people think about, but if you think about the biology of it, it makes sense. So common variation could be, let's say, ethnicity specifying variants. But obviously, if they specify ethnicity, they have a functional effect, right? They may not be causing the disease, but since certain ethnicities are predisposed to certain diseases, you would expect to see that there may be an involvement of common variants in generating disease. Okay, so some conclusions: SNAP scores correlate with the amount of variant effect, and as much as half of the non-synonymous SNPs in healthy individuals are functionally non-neutral. But while both common and rare variants in the genome have effects, and both can contribute to disease. So that's just on the non-synonymous side. So then we wanted to do the same thing with synonymous variants, because as you saw, synonymous variants have an effect, right? And believe it or not, there's almost no tools out there for evaluating synonymous variants because it's not as easy and there is no training data. So there is very limited experimental data that says this synonymous variant has an effect on the resulting protein, right? So, but they can have an effect. They can have an effect on transcript stability. They could have effect on splicing. They can have effect on translation and co-translational folding. So there are many things that could happen. So we set out to develop a method to predict the effect of synonymous variants, but we had no data. So what did we say? We're going to use this vast laboratory of the human evolution in order to try to evaluate the variants. So we collected 4 million observed variants from the Nomen database and also 4 million singletons. So singletons could be sequencing error or real variants. So we sort of just left them out of consideration. And then we artificially generated the remaining 57 million variants that are possible for the human transcripts, but are not observed, right? So our assumption here was that we're going to see an enrichment of neutral or kind of neutral variants in the observed world and an enrichment of deleterious variants in the artificial world. And now I understand that there are variants that have an effect in the observed world, and there are lots of variants that are not observed that have no effect, but the enrichment, I suppose, is going to persist. So what we did is we focused on the observed and the artificially generated variants, and we asked the first question, can we identify any variants in the observed set which we cannot imagine seeing in observations, right? Ever seeing in people. And we did some applied positive unlabeled learning to do this. And this is how it works. You have your observed variants, you select a sample of the generated variants, and you train a model to recognize something as being observed versus not observed. And then you run the entire collection of the generated variants to make a prediction of observed versus not observed on this in this model. And so here, what's going to happen is some are going to be predicted observed. So we're going to say we have the potential of seeing this, we just haven't seen them yet. And we take them out and we do this again, right? And we keep doing it until the model converges. And that happened after about 72 epochs or so. And what we figured is there are 6 million variants, which we termed unobservable. So we cannot describe them using our features, which may be a problem, or we cannot sequence them, or they're so super deleterious that they're never going to appear. So these 6 million, we can't really pass any judgment on because they're not observable. Now, of the remaining ones, we asked, can we differentiate the observed from the not seen, right? So it's now a classifier that differentiates the two rather than saying one is positive and the other is unlabeled. So we trained this classifier and actually it was able to do fairly well. You could see that this is the distribution of observed scores and this is the distribution of not seen scores. And what we then said is, okay, well, this model distinguishes observed and not seen, but now let's see how well it does for common variants and for curated, so we manually picked it out from literature, effect variants, so synonymous variants that have an effect. So these were left out of training, so there is no overfitting of this model to fit these. And what you see very clearly here is that the curated effect variants tend to fall way higher than the common variants. But remember, the effect variants are actually observed. So if they were neutral, they would be here, but they're not, they're up here, which suggests that our assumption that not seen variants are enriched in non-neutral variation is correct. So what we decided to do next is then improve our training to focus more on effect versus no effect. And we selected these cutoffs, so at the medians of the two. But we only took the data from the observed and the not seen, and we trained another model, the final model, to recognize effect versus not effect, as opposed to observed versus not seen. And here's the performance of this model on a data set of variants that we've painstakingly extracted from ClinVar. And this is the two other methods that are capable of annotating synonymous variant effect. And what you see here is this is the default cutoffs. So CAD predicts both of these, both benign and effect variants in our set below the cutoff, which means it predicts them to be benign. Dan predicts them above the cutoff, which means it predicts them to be deleterious, but both of them. You can tell the difference between distributions, but they're all predicting above or below the cutoff. And here's our predictor, SynVap. And what you see very clearly here is that the effect variants score higher on average than the benign variants. So we trained SynVap to predict synonymous variant effect without a training set of synonymous variants, but it works. So this is kind of cool. This is not unsupervised learning, and maybe we should try that in the future. But for now, this is a method that works, and hopefully we'll be able to provide it online shortly. Alla, how much time do I have?
12 minutes.
Oh, okay. Well, okay. So I'm gonna very quickly go through one last thing, and then I'm gonna stop. So people tend to think of variants as the one thing that causes disease. Like, this is the last variant, that's the pathogenic variant, that's the thing that caused disease. As this X-ray demonstrates, the back of the camel is already broken before that last variant lands. So the idea usually is that there is a collection of variants that gives you the disease. It's not one variant that is the pathogenic one in most cases. So we built a model called AVA-DX, which stands for Analysis of Variation for Association with Disease. The AVA-DX takes variation data from patients and controls, and it both identifies the genes or the pathways that could be involved in the disease, and it is a fairly accurate predictor of the disease risk. So here we looked at Crohn's disease because that's the data we had. And I want you to notice that it's only 111 people. So as compared to these large scale genome-wide studies, this is not much. So we described variants in terms of the gene function. So remember how I said SNAP is a predictor of effect? So now we can do that. We can apply SNAP for every variant to the gene function. And then we can sum or multiply. So in this case, we summed the scores of the variants modified by the heterozygosity at that position. Right? And so that way we could represent every person as having a certain function of every gene, and the vector of genes is unchanged. So this is all the genes that you carry around. So your gene 1, for instance, in subject 1 could be completely functional, and gene 2 completely dysfunctional, and this person has Crohn's disease. So that's the way that we represent people. So now what we can do is we can apply feature selection techniques to identify the genes whose functionality is actually well correlated with the effect on the entire person. So Crohn's disease versus healthy control. And what we found is that these genes tend to interact, and they interact in multiple ways. So for instance, here, all the genes that have a black name over here are the ones that we identified, while all the genes that have a purple name are ones that we haven't seen, but are identified by our pathway analysis over here. And what you see here is that all of these black genes tend to be regulated by these same 2 transcription factors. Now, we didn't identify these transcription factors, but these genes were correlated, were related by the fact that they're regulated by these transcription factors. And so that suggested that these genes are involved in a similar pathway or the same pathway. And by the way, here's NOD2. That's the infamous Crohn's disease gene that everybody says is the main driver of everything. We also identified NOD2. And another way that they interact is in molecular pathways. So this is basically protein-protein interactions versus biochemical interactions. And if you look at which pathways these are, what you see is an enrichment in some pathways which are indeed related to Crohn's disease. So let's say apoptotic cleavage of cell adhesion proteins, which is a known problem for Crohn's disease. Right? So AvaDX was able to accurately identify CD patients. But if you wanted to figure out whether a person is predisposed or is at risk, you would need to use a set of people that weren't used for training, right? So you would need to have to do this for a new person coming in. And what's important to remember here, this is RNA sequencing problem. So you have batch effects. So if someone sequences today versus tomorrow, they can get very different results. But believe it or not, there is a batch effect in just genome sequencing as well. So it's a very different kind of batch effect, but a lot of there is a difference, and there are problems to be had. So if you wanted to evaluate the genome sequence of a person that was not used in training, you would need to correct for batch effects first. And actually, we used a method developed by Evan Johnson over in your guys' territory in order to correct for batch effect. We misused it because it's meant for RNA-seq. But since we're looking at the functions, we felt that we were all right with doing that. So I'm just going to skip the slide, but I wanted to make sure that you guys know that his tools are being used very well. And the idea is here that we were able to evaluate our own data set after batch effect correction, obviously. So that's not a problem. So you could see this is the score on the y-axis, and these are sick people and these are healthy people, right? So you could see that the sick people are on average scoring higher. At above a cutoff of 45, you will see that there's only sick people. But now I can add another set of people, and we're still doing good. And I can add one more set of people. Now this set is a fairly large set, right? So we're talking about nearly 3,000 Crohn's disease patients versus 635 healthy controls from GTEx data. And you could still see that this cutoff that has not changed is the same cutoff that tells you about Crohn's disease risk. So this pipeline was used on Crohn's, but it's actually a pipeline for any disease. So as long as you have data, you're welcome to try it out. It has machine learning components that you can change, quality control components that you can change, variant and gene scoring components that you can change, and feature selection components that you can change. And that is optimizable to whatever method you want. What I find curious is this part, because it could also serve as a robust platform for evaluating variant scoring. But it all sits on a Docker image and it's available. Feel free to change it, feel free to use it. I'm just going to show you very quickly. This is me looking at Tourette data. We're able to differentiate Tourette's patients from controls, even when people are related, so in trios. When we look at the pathways that show up, the ones in red are actually known to be related to development of Tourette's. We are also looking at to predict embryonic aneuploidy from the mother's genome, which is a tough task, right? Because there is also a dad involved somewhere there. But the point is that we were able to do that poorly, but we were able to do that. And the genes that we identified were actually heavily involved in cancer pathways contributing to genomic instability, which to me indicates that genomic instability in embryos is also the issue for aneuploidy. Okay, so that's it. Disease pathways are consistent across patients. Batch effects complicate prediction. Genome-protein interactions in known pathways help validate prediction results. And AvaDX may be used as a platform for evaluation of variant effect prediction methods. So the bottom line, we design computational tools to answer questions about cellular molecular mechanisms, however applied. But we also do things in environmental microbiome and bioremediation. as well as the evolution of life on Earth and other planets. And of course, I don't do anything. Everything is done by my lab, and I just talk about it. And so thanks to them, thanks to the funding, and thank you.
Thank you, Yana, for this great talk, very inspiring. So I would ask people, I muted everybody, I would ask people to unmute themselves when they try to ask question. I hope it works. Nelson? It doesn't work? I don't know what to do. Because I was asked to, yeah, I don't know how to unmute people, so I'm not very, all right, wait a second.
I can turn on the chat and read your questions.
Allow participants to unmute themselves, okay.
All right, great, Yana, that was fantastic. I
Thank
had
you.
a question about your populations that you were analyzing both in the microbiome and in the SNP variation. How racially diverse are those populations, or are they not diverse at all? And if you looked at different racial populations or underserved racial populations, would you see very different results, both at the microbiome level of characterization as well as the SNP variation?
So that's a very good question. For the microbiome level, their ethnicity or race, in my opinion, doesn't matter as much. It matters, but it doesn't matter as much as what they eat. And there is very clear signatures of the type of food that they consume. But in Crohn's disease analysis, we only had one family, so they were all related. In the type 2 diabetes, this was a Chinese hospital study, so they were of one ethnicity. I don't remember what else I did. With Prader-Willi syndrome, that was also a Chinese hospital study. So we haven't really looked at diversity there. But for AVA, we clearly have a problem of ethnicity. And for every one of these, the first part of the pipeline is the quality filtering, which is basically is going to tell you if people are A, related, and B, of what ethnicity they are. And so then it's on you to filter out the individuals according to, to what you believe is the correct set to look at. So for Crohn's disease, again, this was in collaboration with Institute in Kiel in Germany. They were actually all Europeans, so there was no question about that. Although I was a little surprised, Kiel is a very big mixing ground at support, so I was a little surprised to see that. But they were rating as Europeans. For Tourette's people, we had a huge variety. So we had to build 5 different models in order to be able to understand what's going on. And these were indeed separate models that didn't work well across ethnicities.
Cool.
More questions? I have a question about synonymous variants.
Yeah.
Sometimes, well, I know little, but I know of synonymous variants that are not, if they correlate with disease, but not because of themselves, but because of their linkage, the equilibrium connection to something that is important. Are you including this information in your models?
No, so these are not disease predictions. These are functional effect predictions.
Functional effects. So that variant then will be really not functional, but another that it linked to perhaps would be.
So we did not address linkage disequilibrium at all, so I can't give a comment on that.
Okay, all right, thank you.
So the misleading portion here is ClinVar. So it usually is clinical data, but we actually manually curated the variants. So we looked for literature references for why it's that.
Okay. Anybody else?
I have another question about the looking glass deep learning on the microbiome. When you go to the machine learning, you're now just looking at the base sequence, the nucleotide sequence.
Yeah, yeah.
Is that considering the triplet code, or is it just reading the
So
sequence without triplet code modeled in it?
So the deep learning model, this thing, doesn't care about anything. It's just a way of representing the letter code in the context of the sequence. So basically, if you were trying to build a model of language, which people do consistently, so think about Siri and Alexa, this is what's happening with them, right? They are trying to build models of language where they recognize word order. But if you were trying to read just words, you would mask out a nucleotide, a single letter. And then you would say, can I predict that letter? So I know I have an alphabet of 4. Can I make a prediction of what is the next letter? And I will make that inference on the basis of what I have seen in the sequence up until then, right? So the idea is to train a model to speak, right? You want it to start understanding the language. So we are replacing the letters by these embeddings or these multidimensional things, right?
Yeah.
I don't know how to explain that. So the alphabet is a multidimensional thing that we just tend to use as is. That's a representation. And if you could think of a series of numbers that represents that instead of letters, I think that's as close as I can get in terms of explaining what the aim is. But once you've learned the language, then you can go on to ask very specific tasks, to ask very specific questions. So does this encoding stand for function X?
Yeah, I'm surprised that you can do that without considering the triplet code, considering how universal that is.
So I spent my entire scientific career training models to do things on the basis of things you expect to matter: secondary structure, solvent accessibility, I don't know, biochemical consistency of the substitutions, and all of a sudden this thing comes in, which is just sequence based and has enough meaning in it to deconstruct whatever the language is. It's super powerful. It's amazingly powerful. And that's why it's so popular right now, because you can answer all kinds of questions.
Wow. Okay, great. Thank you.
Yeah.
More questions? No? If there are no more questions, I would like to thank Yana for wonderful talk and discussion. And yeah, we hope that we would have a chance to see you again, perhaps, talking to us. Thank you.
Thank you. Thank you for paying attention to me.
And your next meeting at 3:15 with Andrew. I hope he's around. Yeah, he's around.
Okay, I will join.
Yeah, there is a link, and you should be able to. I will monitor to see that you joined and everything, but I think everything should go well. All right.
Thanks so much for hosting me.
Yeah, it was wonderful. I learned a lot. I learned the implications, not the how to do it, of course.
It's okay. I mean, the important thing is the questions, the technical stuff we can do. That's the. Yeah. What are the questions that one can ask? That's the big deal.
That's it. Yeah, of course. All right.
Okay, cool.
Have a good break, and
thank you so much for hosting me. Bye.
Bye.